Hi everyone, I'm Candace and today I'm making kimchi, but with a little twist, a science twist. We're going to explore how microbes make this classic Korean dish not just tasty, but also alive. Let's get into this recipe and into the biology and science behind it. Alright, now you can see that I have all my ingredients laid out to make this successful kimchi snack. Let's go over it. I have cabbage, onion, garlic, radish, green onion, ginger, carrots, the stuff for the paste. I have sardines, fish sauce, spicy chili oil, sugar, soy sauce, and salt. All right, now you can see I have my cabbage, my salt, and the sugar here. First, what I'm going to do is sprinkle around some sugar around the cabbage. I'm going to do this with a couple tablespoons. And then next, I'm going to grab my salt and I'm going to do the same thing. Sprinkle around some salt around the cabbage. Doing this helps draw out water and creates a salty environment. But let me tell you what's really happening here. We're setting the stage for microbial fermentation. Salting keeps harmful bacteria away and gives good microbes like Leuconostoc mesorotes a chance to shine. This lactic acid bacterium lives on the surface of vegetables and thrives in a salty, low oxygen environment. Look at all this salt and sugar on this cabbage. Mix, 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 mix. 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 So here I have my cabbage all ready to go and my chili paste all ready to go. Now I'm mixing everything and packing it tightly into a jar. This helps push out air and keeps it anaerobic, perfect for lactic acid bacteria. Over the next few days at room temperature, fermentation will take off. The pH will drop, which preserves the kimchi and adds that signature tang. So here's the science. L-mesenterides uses enzymes like lactic dehydrogenase to convert glucose into lactic acid. This makes the environment acidic, which stops harmful bacteria and lets the good ones take over. Some species even stick around the final product, which is giving us the probiotics that support a healthy gut biome. And now you can see me mixing it up completely, and you got to make sure it's all coated and covered and ready to go. And after a few days, I'm going to stick this in the refrigerator and it'll be ready to go. And here we have it. The kimchi has been fermenting for a few days, and now I can put this in the fridge. Fermentation slows down, but the flavor keeps developing. The microbes may be tiny, but their impact on our food and our health is massive. So this is kimchi, which has so many microbes in it. I can't wait to give this a try. Hope everyone enjoyed the video, and thanks for watching, and happy fermenting.
We'll be discussing sourdough fermentation, focusing on the key microbe Lactobacillus sanfranciscus. Sourdough has a rich history dating back to the 2nd millennium BC in Egypt and the 1st millennium in northwestern China. Lactobacillus sanfranciscus is the only microbe that can be found in sourdough. Sourdough starters are leavened by a combination of wild yeast and lactobacilli, typically in a ratio of 1 to 100. Most common yeast species involved in Saccharomyces cerevisiae, also known as baker's yeast. Bread production involves the mixing of flour, water, and sourdough starter. Sourdough fermentation is a process that transforms white or rye flour into bread by leveraging the metabolic activity of lactic acid, bacteria, and yeast. Depending on the region and culture, different flours such as wheat, barley, rye, and maize have historically been used. Lactobacillus sanfrancis is a gram-positive, slender, rod-shaped, non-sporulating, and non-motile bacterium. It is heterofermentative, meaning that it ferments its own sugars to produce multiple products in mainly lactic acid, carbon dioxide, and ethanol. Yeast as a starter cannot metabolize maltose, while Lactobacillus sanfrancis depends on it, forming a symbiotic relationship between the microbes. During fermentation, the microbes in the starter consume the sugar and produce lactic acid, which gives sourdough its tangy flavor. Yeast fermentation occurs during the anaerobic conditions, typically when the pH range is 4 to 5. The bubbles in the sourdough are the result of carbon dioxide production. As the bread bakes, Lactobacillus sanfrancis begins to die off and when the internal temperature reaches approximately 60 degrees Celsius. However, lactic acid and other byproducts is produced remains in the bread, contributing to its distinct flavor and texture. Sourdough fermentation is a traditional practice that has been passed down through generations and is still enjoyed around the world today.
Hello everybody, I'm going to be talking about baked kimchi and the role that Levulactobacillus brevis has in its fermentation process. So kimchi is a Korean-based dish. It does consist of a lot of fermented vegetables. There are hundreds of different versions of it, like bak kimchi, pak kimchi, and other countries have similar dishes like pao chai and tsukemono, but the one I'm talking about today is napa cabbage kimchi, aka beichu kimchi. So a lot of traditional ingredients include salt, a spicy mixture, and other vegetables. The main process it goes through is fermentation in aerobic environments, where the lactic acid bacteria come into play, especially the Levulactobacillus brevis that we will be discussing today. From the domain of bacteria, and its main function is to ferment carbohydrates. Some characteristics is that it's gram-positive, it's rod-shaped, and it's usually single or in pairs. Some metabolic involvements include the fermentation of sugars into lactic acid like glucose, fructose, and maltose. It resides naturally on vegetables and it likes a temperature of 15 to 20 degrees Celsius, a pH level of about 5.5 to 6.5. It is heterofermentative. It likes substrate level phosphorylation where pyruvate is the final electron receptor. It turns glucose into pyruvic acid/pyruvate and then into lactic acid. It's not hemofermentation because other byproducts like carbon dioxide and ethanol are produced, and because it goes through phosphoketolase phosphorylation. It produces lactic acid via lactic acid bacteria, and this happens when sugar is broken down, and it results in a lower pH and helps kill the bacteria. In both small and large scale production, they're both cultured in nature and they're both collected in those fermentation tanks or jars, and you could also process them by harvesting or by getting a concentrate. During this whole process is alive in the brine, the fermentation, the consumption, and in the digestion. In the body, it's used as a probiotic to help with gut and boost immune system, and it has been shown to lower inflammation in the intestinal cells. Not a whole lot of GMOs are used because everything happens pretty naturally. It's only used when going into experimental stuff, and it's also trying to see if it could be a possible biofuel precursor with biotechnology. Anyways, thank you for watching.
Soy sauce production is a fascinating example of food fermentation, a process driven by the metabolic activities of diverse microorganisms. This process transforms simple ingredients into a complex and flavorful condiment. Soy sauce is known to be Asia's most staple ingredient. The process begins with a mixture of soybeans and wheat. This mixture is then inoculated with specific molds, primarily Aspergillus oryzae and Aspergillus sojae, to create koji. These molds are aerobic filamentous fungi that thrive in this environment. Aspergillus oryzae and sojae are characterized by their extensive network of hyphae which penetrate the soybean and wheat mixture, enabling them to efficiently secrete a wide range of extracellular enzymes. The koji is then mixed with a brine solution to form a mash called moromi, initiating a secondary fermentation involving a complex community of microorganisms. The halophilic yeast Zygosaccharomyces rooksii plays a crucial role in this stage. This yeast is highly tolerant to salt concentrations, a key characteristic that allows it to ferment sugars produced by Aspergillus molds into alcohols and various aromatic compounds, contributing significantly to the final flavor profile. The lactic acid bacterium Tetragenococcus halophilus is also essential. This bacterium, like the yeast, is halophilic, enabling it to thrive in the high salt environment. Tetragenococcus halophilus produces lactic acid, which lowers the pH, inhibiting the growth of undesirable microorganisms and contributing a characteristic tangy flavor that balances the overall taste of the soy sauce. The raw soy sauce is usually pasteurized to stop any further microbe action and make it stable. It might also be filtered to make it clearer. So in the end, tiny living things like molds, yeast, and bacteria work together to turn simple stuff into tasty soy sauce.
A trendy health food, homebrewers dream, and fiddling acquired taste. Love it or hate it, kombucha is inarguably a bubbling ecosystem of symbiotic microorganisms that come together to form a unique, and in my opinion, delicious beverage. The controversial drink originated in China around 220 BC, but you can find it at pretty much any store nowadays. Lots of people drink it for its famous probiotics, but not many people actually know what's going on in there. The drink is made from a tea base, usually black tea, but others can be used like green or oolong. This is mixed with some sugar and then you plop in a SCOBY, or a symbiotic culture of bacteria and yeast. This gelatinous blob is exactly what it sounds like, specifically made of acetic acid bacteria, lactic acid bacteria, and yeast. The bacteria that give kombucha its signature acidic and vinegary taste is the acetic acid bacteria, which are largely made up of the genus Comagotibacter. This also gives it its jelly structure. These are gram-negative, rod-shaped, obligate aerobes that can grow in single bacteria, pairs, or chains. They work with other SCOBY microorganisms to ferment the sugars in the tea to make kombucha. First, the yeast hydrolyze the sugars to make smaller sugars, then ferment them to pyruvate, then to ethanol and carbon dioxide. That's what gives kombucha its bubbly bubbles. Then the acetic acid bacteria take the ethanol and turn it to acetic acid, giving it its signature flavor. You need to make sure that the drink ferments for long enough to be kombucha, but not so long that it tastes like straight vinegar. Once you made your drink, you can just reuse the SCOBY to make even more.
I did microbes in my food and I chose to do sourdough bread. So, sourdough bread goes through alcoholic and lactic acid fermentation. To make this, you need flour, water, and salt. A key component that you'll need to make is the starter. So, you'll add water and flour and leave it room temperature from anywhere from 2 days to a week. The natural yeast and lactic acid bacteria will colonize into the starter. You want to make sure that you have warm water so that the bacteria is able to be attracted into your starter. So the yeast and bacteria will colonize in there to create rising. So once your starter is at peak of its log phase, you're going to want to then add in water, salt, and flour to help activate the metabolic reactions. Adding the warm water will continue to populate and keep the starter active, and the salt will also control the yeast activity. So wild yeast gives the bread its airy texture, and it is responsible for fermenting sugars from the flour into CO2 and ethanol. So the flavor enhancement can be affected in many different ways: the balance of ingredients, temperature, or how long that you let your starter ferment for. Alcoholic fermentation is when the yeast consumes sugars that are present in the flour and produce carbon dioxide and ethanol as byproducts. This is what gives the dough the airy texture and the bubbles in it. Lactic acid bacteria fermentation is a lactobacillus species that is common in lactic acid, and this is when the pH gets lower than 4.2 that then makes this a pathogen-free zone and creates lactic acids and acetic acid, and it makes it safe for us to eat. Also, when you put it through the oven, it will then kill all the pathogens and make it safe to eat and no longer metabolically active. So lactobacillus is a microbe that is rod-shaped and it is gram-positive. And putting these all together is what gives you the delicious bread as you know it and gives it sour taste, airy texture, and makes it very different from other breads that you may buy at the store. And these are my works cited.
You may not know it, but there is an invisible war being fought around you every day. The war on microbes. Many have fallen victim to the harmful anti-microbe propaganda, but the truth is that we would not be here without the millions and millions of tiny microbial organisms that call us home. What if I told you that some of the most important neurotransmitter factories for your brain aren't in your brain at all? They're in your gut, the most microbially dense part of your body. And one of the most powerful? A tiny microbe called lactobacillus. Lactobacillus is part of your gut microbiota, a massive community of bacteria working around the clock to keep you healthy. It's found naturally in fermented foods like yogurt, kefir, sauerkraut, and kimchi, and helps the gut digest food by lowering gut pH and inhibiting harmful pathogens. But here's the real twist. Lactobacillus also produces neurotransmitters, chemicals that directly affect your mood, memory, and even motivation. It synthesizes GABA, a calming chemical that reduces anxiety and assists in the production of dopamine, the neurotransmitter responsible for feelings of reward, pleasure, and motivation. This is all possible because of the gut brain axis, a communication superhighway between your intestines and your brain. When we overuse antibiotics or obsess over killing germs, we don't just lose pathogens, we lose allies like lactobacillus, and that loss may contribute to rising rates of depression, anxiety, and even addiction. Protecting these microbes isn't just about gut health anymore. It's about protecting our mental health as well. To protect our future, we must start by protecting our smallest allies. So next time you think about taking care of your mind, start by taking care of your gut.
My name is Loy Lu. Today I will share how tiny microbes have made one of the world's most popular foods, bread. Bread is enjoyed across culture. Most traditional breads depend on microbial fermentation to rise and develop flavor. It starts with the simple ingredient flour, water, salt, and yeast. With flour provides starch for yeast to break down. After mixing and kneading, yeast ferment the dough, producing carbon dioxide that gets trapped and makes it rise. The main microorganism is Saccharomyces cerevisiae, or baker yeast. It's a round, single-celled fungus that prefers fermentation when oxygen is low. Yeast is grown in tanks with molasses and processed into fresh or dried forms. Most yeast die during baking, but their byproducts stay, which adding flavor and texture. Through alcoholic fermentation, yeast converts glucose into carbon dioxide and ethanol. The carbon dioxide from bubbles that lift the dough and ethanol adds flavor before evaporating. This microbial action turns heavy dough into soft, fluffy bread. Modern bakeries use selected yeast strains for faster rising or special flavor. Without yeast in fermentation, bread wouldn't be the same. Microbial metabolism gives bread its rise, aroma, and texture.
Hello, friends. My name is Lexi. I'm going to go over the microbiome I wanted to present to you guys, which is the Bifidobacterium longum. Here are a couple learning points that I wanted to walk us through during this presentation. The first one is the brain-gut axis, which is a communication highway between our gastrointestinal tract and our brain. Through a network of nerves, hormones, and immune pathways, signals from our gut can directly impact our mood, memory, and even our stress levels. Bifidobacterium longum plays a huge and key role here by producing compounds like GABA, a calming neurotransmitter, which helps regulate emotional health. For the gut ecosystem, our gut ecosystem is made up of trillions of microorganisms living together, including bacteria, fungi, and viruses. Bifidobacterium longum is a native member of this ecosystem, helping to maintain a healthy gut lining, preventing from harmful bacteria from overgrowing, and even assist in nutrient absorption. It thrives best when the gut environment is balanced and nutrient rich. Bifidobacterium longum is considered beneficial or a good bacterium. He's a good guy. It's one of the first species to colonize our intestines after birth, and it helps train our immune system to recognize friend from foe. In return, we're provided a safe, nutrient-filled place to live, a perfect example of a symbiotic relationship where we both and the bacteria benefit. Some of the impacts of poor microbiomes: when our gut microbiome becomes imbalanced, whether from stress, antibiotics, or poor diet or illness, we lose key players like Bifidobacterium longum. This can lead to gut inflammation, poor digestion, and even contribute to the anxiety and depression, showing just how important these microbes are for our overall health. For probiotics, one of the ways to restore balance through probiotics are live microorganisms that confer health benefits. Supplements or fermented foods containing Bifidobacterium longum can help replenish its levels. Supplements replenish these healthy microbiomes and support the gut-brain axis for better mental and physical well-being. In closing, Bifidobacterium longum isn't just a passive resident of our gut, it's an active partner in protecting our health, and both physically and emotionally. Supporting our microbiome means supporting ourselves. Thank you for listening.
Hello, my name is Grace Lane, and I am going over Penicillium camemberti, which is a fungus that makes soft rind ripened cheeses such as camembert, brie, and a lot of other delicious things. So this is my favorite cheese ever, and it is made in France, and they use P. camemberti, and they spray it on the rind of the cheese so it gets established and makes a mycelial cover around the cheese and gives it its unique taste. So when P. camemberti is on this rind of the cheese, it metabolizes lactate into CO2 and H2O, and this enzymatic activity causes an increase in pH on the rind, which causes the minerals in the hardened cheese curd to migrate from the lower pH in the center to the outside rind, which creates that thick rind on the outside and that delicious, creamy, soft cheese on the inside. So P. camemberti is a type of fungi. It has conidial heads, which are also known as fruiting bodies. And they're called conidial heads because they produce asexual conidia spores, and they are aerobic microorganisms. So one of the processes that I highlighted was proteolysis, and this is something that P. camembert does, and it is the process of breaking down proteins into free amino acids. We have a lot of the enzymes that do this in our body, in our digestive system, and in the cheese, the P. camemberti takes the proteins from the milk caseins, and they break them down into free amino acids, which gives the cheese its specific and unique flavor. And the mycelium that covers the cheese allows those acids and broken down lipids to stay trapped within the rind and really give it a unique flavor. So now it is your task to go try some cheese with P. camemberti on it, and let me know what you think.
Hi everyone, my name is Mia Nguyen, and today we're going to dive into one of the strangest and most hyped microbes, cordyceps, also called Đông Trùng Hạ Thảo in Vietnamese. From ancient healers to TikTok trends, cordyceps is claimed to boost energy, heal lungs, and slow aging, and it sounds too good to be real. Đông Trùng Hạ Thảo really just means winter insect, summer grass. It infects a caterpillar in the winter and then grows into a mushroom from its head in the summer. It's pretty gross but fascinating. For centuries, the people in Tibet and Nepal, they risked their lives in the Himalayas to harvest wild cordyceps. It was so prized that during the Qing dynasty, it became a tribute for the king only, value more than jade or gold. Overharvesting led scientists to grow Cordyceps militaris in lab using rice instead of caterpillars. It gives the same powerful compounds without harming nature. This bright orange fungus, it loves oxygen, prefers 20 degrees Celsius, and it likes low light. It is now the top strain used for supplements because it is easy to farm and potent. Inside the fungus, we have structures called hyphae to absorb the rice nutrients, and with oxygen, it turns sugar into ATP, and the superhero molecule is cordycepin. Cordycepin mimics adenosine in your body. It helps reduce inflammation, boosts energy, and even support immunity and brain health. Growers boost cordyceps by tricking humidity, temperature, and even using blue LED light. And by the time it's in your tea or capsule, it's dry and powder, so you're sipping the metabolite, not the living microbes. Ancient Chinese medicine used cordyceps for vitality and lungs, and modern research back its effect on ATP, energy, inflammation, and immune health. So do I believe now? Not really. I'm still skeptical. I believe in eating variety of food will give you the same health benefit, but I think it's fascinating. And this is just a page on what I use, articles that I use reference. So thank you for joining me. Stay curious and stay healthy.
Hi everyone, my name is Anna Geva, and my project is about kombucha. Kombucha is a fermented tea made using a symbiotic culture of bacteria and yeast or SCOBY. Kombucha is made by combining sweetened tea with a SCOBY and fermented it for 7 to 14 days. Yeast converts the sugar into ethanol, and then bacteria convert the ethanol into acid, like acetic acid, giving kombucha a tangy taste. The key microbe I'm focusing on is Commagatiobacter zelenis, an aerobic rose-shaped bacterium that not only produces acetic acid but also creates cellulose, forming the structure of the SCOBY. The main metabolic process I look at is ethanol oxygenation. This process helps preserve the kombucha, lowers its pH, and contributes to its potential health benefits. I chose kombucha because it's a great example of how microbes shape the food we eat, from flavor to function.
Today I'd like to talk to you about Staphylococcus auricularis and your ear canals. Most of us are familiar with the fact that the human body is made up of a mixture of human cells and other cells, mostly trillions of bacteria that co-evolved with us. We think about how taking too many antibiotics can throw off the microbiome of our gut, and we take care to prevent the bacteria in our mouth from forming the biofilms that can cause cavities. But what about our ear canals? It turns out they have a microbiome too. First, a little bit of anatomy. Your ear canal, formerly called the external auditory canal, is about 2.5 centimeters long. It starts at the hole you can see on the outside of your ear and ends at your eardrum. The outer third of the canal is made of cartilage, and it's lined with glands that produce cerumen, also called earwax. The inner 2/3 are made of bone overlaid with a thin layer of skin, which is continuous with the eardrum. Lots of different bacteria hang out in your ear canals. One is Cutibacterium acnes, which is found in the sebaceous glands of hair follicles all over your body. Another is Pseudomonas aeruginosa, an opportunistic pathogen that's the most common cause of ear infections. But I would like to focus on Staphylococcus auricularis. Unlike its more famous cousin, Staphylococcus aureus, Staphylococcus auricularis is rarely harmful, even when given the opportunity. S. auricularis is a gram-positive coccus, generally found in tetrads or pairs. It's a facultative anaerobe that grows in small, white, circular colonies. It's catalase positive. The research we have so far shows that Staphylococcus auricularis is the most abundant microorganism in a healthy ear canal. Researchers aren't totally sure yet what Staphylococcus auricularis does for our bodies. They do know that when someone has an ear infection, the balance of the ear microbiome shifts, and S. auricularis is overcome by large numbers of Pseudomonas aeruginosa and other microbes. So how can you keep the Pseudomonas aeruginosa in your ear canals from overpowering the Staphylococcus auricularis? The best method is to leave your ear canals alone. Earwax has antibacterial properties, and your ear canal is self-cleaning. As earwax naturally leaves your ear, it will take debris and pathogens along with it, helping you stay healthy.
This is a human. If we put on special glasses, it will allow us to see the human microbiota. The human microbiota is the many different microorganisms that live in and on us. They help keep us healthy or can contribute to disease. Let's take the glasses off and zoom into the oral cavity. The oral cavity provides some ideal conditions for fungi and bacteria. To see some of these communities, we will perform a staining procedure. The stain differentiates between age of plaque. Pink shows new growth, dark blue or purple is mature growth over 48 hours, and light blue is over 3 days old and acid-producing biofilm. When kept balanced, the organisms live symbiotically, but once imbalance of the organism is reached, that is when disease can occur. Disease in the oral cavity isn't just cavities, but also has been linked to systemic diseases like Alzheimer's and diabetes. Let's put our special glasses on one more time and zoom into those gums to see a microorganism that contributes to periodontal disease. Porphyromonas gingivalis. Porphyromonas gingivalis is anaerobic, gram-negative, rod-shaped, and non-motile, which means it doesn't move on its own. The reason this microorganism is so destructive is down to its metabolism. Porphyromonas gingivalis breaks down proteins for its energy and carbon. This is what leads to periodontal disease. In conclusion, the human microbiota is amazing. It is still being studied to understand the relationships between us and it. And to help keep Porphyromonas gingivalis in check, don't forget to brush your teeth.
My name is Brian. This is my second time making a video myself because the first one I made went over the 2 minutes, so I'm working on tightening it up. Just to jump right in, our bodies is covered with microorganisms. 1 to 3% of our body weight could be microorganisms at any time, so that means like 2 to 6 pounds of me is not me, which is wild. I chose to focus on the gastrointestinal tract or the guts, and at that point there's 1,000 different species of those microorganisms could exist there, and we each have at least 160 species at any time, numbering in the trillions. I chose to focus on Bacteroides fragilis, which is a rod-shaped, gram-negative shaped bacterium. It does not have a flagella. It has a fembrate, which it can use to attach itself. It's also aerotolerant, which is not like most bacteroids, means it can survive outside of the gut and then cause infections in the body. It can be problematic. It can also, it's essential for breaking down carbohydrates and turning into fatty acids that we use for energy. There are 2 different strains of B. fragilis, actually. The one is safe. One is a little more pathogenic, where it secretes a zinc-dependent metalloproteases toxin, which can cause inflammatory diarrhea and even colitis, which is not what you want. What also is interesting about B. fragilis is that it contributes to synthesizing vitamin K, which I didn't know they could do. So I think I got that in 2 minutes, so that's great. Thanks.
Host to trillions of microorganisms that make up the microbiome. There are as many microbiota as cells in our body. Some are beneficial and some are pathogenic. This microbiome begins forming at birth and continues to grow and change as we do. It's even responsible for the growth and development of our immune system. Each site on the body has its own community of specific microbiota. Inside of the mouth is a diverse variety of bacteria, viruses, yeasts, and fungi. With a relatively neutral pH, a temperature of typically 98.7 degrees Fahrenheit, many structures and crevices in a wet, moist environment, this is an ideal place for microbiota to grow. There's nearly 1,000 various species in the mouth. From Bifidobacterium, a bacteria that's relatively harmless and can even promote good oral health, to Porphyromonas gingivalis, a pathogenic bacteria responsible for local degradation such as gum disease, to systemic illness such as Alzheimer's and heart disease. The teeth are non-shedding, like most of the mouth, which makes it an ideal environment to adhere to. Biofilm is the mesh-like network of microbiota on the teeth. It's also known as plaque. Within the biofilm, we find Streptococcus gordoni, which makes up to 70% of the biofilm. Gordoni is not usually pathogenic, but it's a coaggregate for P. gingivalis. P. gingivalis adheres to Gordoni and can use its lactic acid as an energy source when necessary. But they also have an antagonistic relationship. S. gordoni can reprogram epithelial cells to prevent infiltration of P. gingivitis, thus preserving its host.
My name is Kate Johnson, and I will be reporting on the gut microbiota with an emphasis on lactobacillus. The microbiota of the body refers to all the microorganisms, primarily bacteria, that live in and on the human body. They contribute over 150 times more genetic information than the entire human genome combined. Benefits of a microbiota is to protect against pathogens, aid in digestion, support the immune system, synthesize nutrients, communicate with the brain, and maintain that gut barrier. Here's some healthy do's and don'ts for your microbiota. Fun fact, the gastrointestinal tract alone contains over 100 trillion microorganisms. Here you'll also find 2 different varieties of bacterium found in your gut. Lactobacillus, my main bacteria that I am looking at, is from the phylum Firmicutes, class Bacilli, order Lactobacilles, family Lactobacillicae, and are related to other lactic acid producing bacteria. They're rod-shaped, gram-positive, non-spore forming, and facultative anaerobes, meaning they are perfect at living in that gut. They do not require oxygen. Plays a large role in our microbiota. They're associated with the intestinal tissue, building that mucus up to keep away pathogens. They ferment food and produce lactic acid from carbohydrates and help maintain that acidic environment that pathogens do not enjoy. And they have a number of proteins that help them to adhere to our epithelium, the host epithelium, in order to help the immune response.
Hello everyone, this is The Human Microbiome and Staphylococcus epidermidis by Tristan Lay. So what is the microbiome? The human microbiome is a diverse and complex ecosystem made up of trillions of microorganisms. This includes bacteria, viruses, fungi, and protozoa. These all live in various places such as the gut, mouth, and even skin. These microbes are essential for our health, helping us digest food, even train our immune system, and protect us against harmful pathogens. And yes, I did say skin. On our skin, we have a diverse community of microorganisms adapted to different environments on our skin, like oily areas, dry areas, or even moist regions. These microbes play a delicate balancing act of both competition and cooperation. And when this balance is disrupted, it can lead to skin issues like acne, eczema, or even increased susceptibility to infection. Among these microbes on our skin, one of the most important and intriguing, in my opinion, is Staphylococcus epidermidis, which you can see on your screen. This bacterium is one of the most common residents of the human skin. But the big question is it a friend or foe? While usually harmless, S. epidermidis is what we call an opportunistic pathogen. That means it can cause infection, especially in hospital settings or when it forms biofilms, which are basically sticky protective layers that make it resistant to antibiotics and immune responses. But here's the twist. S. epidermidis is also a protector. It produces antimicrobial peptides that prevent harmful bacteria like Staphylococcus aureus from taking over. It can also help regulate the skin's pH and even supports wound healing. So while it has the potential to cause harm, it usually works to keep our skin healthy and free from more dangerous invaders. So in short, it's both. And just like Staphylococcus epidermidis, our microbiome, when it's balanced, it will serve to protect us, but when that balance is shifted or interrupted, it can cause us harm. Thank you for watching, and I hope you have a great rest of the day. Thank you.
Hello, and welcome to my presentation on the human microbiota. The human microbiota is essentially a community of bacteria, viruses, fungi, and parasites that live mostly symbiotically within the body and assist in a multitude of biological and metabolic processes necessary to sustain life. The microbiome offers many health benefits, including digestion control and nutrient absorption, immune support, vitamin synthesis, and mental health support. The microbiome lives in a fairly delicate balance, as environmental needs must be met to sustain life and colony formation of the microbes within the ecosystem. Some of the factors that contribute to the diversity of the microbes within the biome are genetics, environmental factors, lifestyle choices such as diet, activity level, sleep, and stress management. Looking specifically at the ecosystem of the skin, many microbes have evolved to thrive in its relatively arid and highly aerobic conditions. Areas here can be classified as dry, moist, or sebaceous, and there is diversity within community in all of these areas. Just like in the human microbiome as a whole, there is a combination of beneficial and detrimental microbes within the skin's environment. One of the species that perpetuates here is Staphylococcus epidermidis, a gram-positive, non-motile, non-spore-forming cocci bacteria that forms in clusters and is a facultative anaerobe. The metabolites of this species generally make it more difficult for more virulent species of Staphylococcus to colonize the skin, such as Staphylococcus aureus. But Staphylococcus epidermidis is technically an opportunistic pathogen as well, as its ability to form a strong biofilm has made it difficult to eradicate in immunocompromised individuals and contaminated medical equipment. Many different species within the Staphylococcus genus are essential to maintaining the health and integrity of skin and mucous membranes, and there are many more microorganisms throughout the body that contribute to the biodiversity within the ecosystem that is the human microbiome. Thank you.
In his TED Talk, Jonathan Eisen called the human microbiota a microbial cloud, and instantly I thought of Pigpen from Peanuts, that dusty kid surrounded by a cloud of visible dirt. Our microbial cloud isn't dirt, though. It's trillions of microbes, bacteria, fungi, viruses, even archaea. And like Pigpen's cloud, they follow us everywhere, on our skin, in our gut, our mouth, even the air around us. These microbes outnumber our genes 100 to 1 and play a huge role in our health. They help us digest food, boost our immune system, regulate metabolism, and even affect our mood and body odor. Lifestyle, exercise, diet, and the environment also shape our microbiome. The skin's environment, its dryness, exposure to oxygen and UV light, and even our sweat shapes which microbes can survive there. The skin's exposure to oxygen makes it most beneficial for aerobic and facultative anaerobic microbes. It lacks readily available metabolites, offering limited nutrients compared to other places in the body like the gut. These microbes on our skin compete for nutrients, secrete antimicrobial compounds, and influence and protect the skin's immune system. For example, Staphylococcus epidermidis promotes immune tolerance, and Malassezia species help the skin fight infection and heal damaged cells. But what happens when things get out of balance? Imbalances can lead to infection and chronic skin issues. That's when we see conditions like acne, eczema, dandruff, impetigo, folliculitis, and rosacea. Staphylococcus aureus is a gram-positive, facultatively anaerobic prokaryote with spherical cells that are typically clustered in grape-like arrangements. It tolerates a lot, including salt, desiccation, radiation, and heat, which allows it to readily survive on environmental surfaces and the skin. It's coated with protein A, which interferes with antibody immune responses, and it also possesses a slime layer and can form biofilms. S. aureus can be problematic, particularly when the skin's barrier is compromised. S. aureus can lead to skin disorders like impetigo, folliculitis, and staphylococcal scalded skin syndrome. It's also notorious for causing more severe infections when it spreads beyond the skin, including toxic shock syndrome, pneumonia, and endocarditis. All this is to say, don't make the Staphylococcus aureus living on your skin mad, or you might find yourself in a world of hurt.
The number of microorganisms in and on the human body outnumbers human cells about 10 to 1. There can be a tendency to focus on pathology when thinking about these microorganisms, but for the most part, their relationship to human health is symbiotic. Because they are hard to isolate and culture out in the lab, DNA sequencing is often the best way to learn more about these organisms that make up the human microbiota. The scientific community has come to the understanding that a healthy, hardy microbiome is less about the identity or presence of any one specific microorganism and more about diversity of microorganisms and collaboration between those organisms. This project focuses in on the gut microbiome specifically. Microorganisms here serve a wide variety of functions. Some help regulate the immune system response, some digest and absorb nutrients, some produce vitamins and amino acids, some are protective against foreign bodies and pathogens. While many of the microorganisms present in the gut microbiota are bacteria and viruses, there are also eukaryotic yeasts and fungi present. Candida albicans is the most prevalent of these eukaryotic species. For 50% of the population, Candida albicans is a harmless commensal member of the microbiota existing without causing harm. A diversity of bacteria in the microbiota acts as antagonists to the Candida albicans, keeping it at healthy commensal levels and preventing its overgrowth in most individuals. When overgrowth does occur, Candida yeasts can migrate out of the digestive tract and cause mucosal infection or infection in the bloodstream. Candida albicans is the most common cause of fungal infections around the world. A big part of what makes it so effective as a pathogen is its metabolic adaptability and the fact that it exists in both yeast and hyphae form and thus can form complex biofilms consisting of these two different morphologies, seen in the image below.
Hi everyone, my name is Rashna Chaudhary, and today I will be talking about Staphylococcus. Staphylococcus is a common bacterium that lives in our skin and inside our noses. While it is often harmless, it can also turn into a dangerous pathogen under certain conditions. Staphylococcus is part of our normal skin microbiota. These bacteria actually help protect us by preventing harmful microbes from taking over and by supporting our immune system. Most people carry it without any issues. However, problems occur when Staphylococcus enters our body through cuts and wounds. One strain particular is MRSA, or methicillin-resistant Staphylococcus aureus, especially concerning because it is resistant to many antibiotics. It causes serious infections like pneumonia, bloodstream infections, and surgical wound infections. Working in a hospital, I have seen firsthand how important infection control is. Simple measures like hand hygiene, using gloves, cleaning surfaces, and isolating infected patients makes a big difference in preventing the spread of staph infections. Staphylococcus is both a friend and a foe. By understanding how it works and following the proper infection control practices, we can protect ourselves and others. I'm leaving you with this quote: we coexist with microbes daily. Understanding them is the first step to protecting ourselves. Thank you for watching.
The skin microbiota refers to all of the organisms, the microorganisms that are found on the human skin, namely the epidermis, which is made of these dead keratinized cells which provide these microorganisms very little nutrients. Not only that, but the skin is slightly acidic, which is actually the body's first line of defense against microorganisms. So it's uncomfortable for a lot of organisms, yeah. But the ones that have adapted to this acidic environment have also adapted to make use of the very little resources available, not only being able to exist on the skin, but actually helping humans by working together to kill off harmful pathogens that might show up on the skin. These helpful microorganisms, they live in the folds of the epidermis, mainly bacteria, fungi, sometimes viruses, but they actually play a very vital role in protection from harmful pathogens. And they work together, they use cell signaling to each other to, help each other know when there's a harmful pathogen that needs to be killed. And one such harmful pathogen that I'm going to be looking at today is Staphylococcus epidermidis. Now, this is actually a organism that's found on the skin normally in small amounts and is pretty harmless, but it is opportunistic, however, and so if given the chance or if, colonized enough in the skin, it'll burrow down into the skin where there's a blood vessel. It'll then inject microbes down into the skin, straight into the bloodstream, and then colonize as this sort of, this kind of rash, on the skin, producing biofilms to nestle it deeper into the skin, protect itself. It's nasty, but in harmful amounts, it's pretty good, and just stay away from contaminated needles, that's for sure.
Hello everyone. If you couldn't tell already, I'm holding up 2 tooth models, and based on that information, you can probably guess what my topic of discussion is going to be about. Today, I'm going to talk about the important role that microorganisms play in our bodies, and specifically our mouths. And we hear it all the time that bacteria are all around us, and it's true. We have tiny microorganisms floating around in the air that are living on our skin and inside of our bodies. Together, they make up what is called our microbiota, which includes not just bacteria, but also viruses, fungi, and archaea. They help keep us healthy, but our microbiomes can also be disrupted by things like antibiotics, medications, stress, and even through lifestyle choices. And our mouths are a great example of how complex and important these microbial communities are. Our mouths are warm and moist places full of nutrients, housing over 700 species of bacteria. And bacteria like Streptococcus salivarius help keep harmful bacteria like Streptococcus mutans under control. But if our oral microbiome gets thrown off balance, often referred to as dysbiosis, harmful bacteria can take over, which can then lead to cavities, gum disease, and other underlying issues. And Streptococcus mutans, it's a major cause of cavities. It feeds on the sugar from our food that we consume and turns them into acid. Then the acid then erodes the enamel on our teeth, eventually leading to tooth decay. And S mutans, it's a gram-positive facultative anaerobe that thrives when our diets are high in sugary fermentable carbohydrates. And in conclusion, keeping a healthy balance in our microbiome, especially in our mouths, is crucial for preventing issues like cavities and maintaining your overall health. Thank you for listening.
Did you know that your body is home to trillions of microbes? This is called your microbiota. It contains bacteria, fungi, viruses, and even archaea. Most of them are super helpful, especially in your gut, your skin, and your mouth. Your gut is like a city of microbiomes. It's warm, moist, nutrient-rich, and full of life, kind of like the sauna. Good bacteria in your gut helps digest food, boost your immunity system, fight off infections, and also help influence your mood. When your gut is healthy, you're healthy too. A balanced microbiome can help fight against obesity, diabetes, heart disease, and even cancer. But things like antibiotics, pesticides, stress, and poor diet can really mess up the balance. Antibiotics can especially kill good bacteria, not just the bad ones. This leads to a problem called dysbiosis, which is an unhealthy microbiome. One of the troublemakers that can take advantage is Clostridioides difficile, formerly known as Clostridium difficile, or C. diff for short. It's a spore-forming, toxic-producing bacterium that normally stays quiet, but when antibiotics wipe out all the good guys, C. diff can cause some serious gut problems, like diarrhea and colitis. Keeping your microbiota balanced with healthy foods, less stress, good sleep, and smart antibiotic use is key. A strong gut means a strong, healthier you, even for your brain and your mood.
The human body is a bustling ecosystem home to trillions of microorganisms collectively known as microbiota. Among these diverse inhabitants, bacteria play crucial roles, some beneficial, other potentially harmful. We have microbiota living in our gut, our mouths, and our skin. Today, Our topic focuses on one such bacterium found on the skin called Corynebacterium jacium. So what is it? Corynebacterium jacium is a gram-positive bacterium, typically rod-shaped and non-motile. It is a normal part of the human skin flora, living in warm, moist areas such as the groin, axilla, and perirectal area. Here you can see it on blood agar, typically appearing as small, non-hemolytic, grayish-white, and glistening colonies. They are often described as having a metallic sheen. While often harmless, C. jacium has notable characteristic. It's frequently resistant to antibiotics, including vancomycin and penicillin. The resistance complicates treatment and makes infections harder to control, especially in a hospital setting. For individuals with diabetes or kidney failure or those undergoing invasive medical procedures, C. jacium can exploit their weakened defenses and cause serious infections. This is treated with antibiotics. However, antibiotic overuse could disrupt the delicate balance of the skin microbiota, leading to an overgrowth of these pathogens. Effective hand washing and general sanitary practices is a must to aid in prevention of these little microbes. Fortunately, scientists are exploring C. jacium's genetics to understand its antibiotic resistance. Deciphering these mechanisms is crucial for improving clinical practice and developing ways to combat this seemingly benign yet problematic pathogen. Thank you so much for watching.